Ever wonder why everyone is getting fat and sick? We've heard a lot of boring and uninspired explanations, but you'll want to watch this video for some fresh new ideas and a lot of interesting research that is going to give a much more definitive answer. First, let's go over some incomplete ideas of what dietary factors cause metabolic illness, starting with the classic eating too much argument, which is people eat too much because food is easy to get, the decision to eat too much results in getting fat, being fat causes chronic diseases like diabetes, heart disease, hypertension, fatty liver, etc. And getting chronic diseases without being fat is due to bad genetics. The brilliant solution is to not eat too much. According to this model, we just have to do our best to restrict calories and hope nothing bad happens. Unfortunately, there are many important questions that this argument doesn't do a good job of answering, a few of which are as follows. Why do people eat much more on junk food diets? Why don't humans or other animals overeat on nutritious diets, even when food is unlimited? Why do overweight people have larger appetites? And why do many people who are not overweight still develop chronic disease? And why do they still improve with dietary changes despite not being overweight? Other incomplete ideas are centered around carbohydrate restriction and usually go something like this. Eating too many carbohydrates causes blood sugar to rise rapidly. The rapid rise in blood sugar causes high secretion of a hormone called insulin, which helps to store glucose, aka the sugar, in cells and slows fat burning. Finally, because of the crash in blood sugar, hunger is increased, and this cycle, when repeated, causes cells to start resisting insulin, which means glucose cannot effectively get into cells. Weight gain happens because of slowed fat burning, and insulin resistance increases, causing chronic diseases to develop. It's true that insulin resistance is tied to a lot of chronic diseases. There's a mountain of research supporting that. But in this case, the cause of insulin resistance is misunderstood. So while this argument is on the right track in some ways, there are still questions that cannot easily be answered, like, why do some people's metabolic health improve on high-carb diets based around whole foods? Why does smoking cause diabetes? Because tobacco is not a carbohydrate. Why is ultra-processed food causing worse metabolic health and increased hunger versus whole foods with a similar amount of carbohydrates? And why does research consistently show honey to be healthier than sugar, despite honey being almost pure sugar? The goal here is to do a much better job answering these kinds of questions that the other ideas can't. The first clue is an underappreciated hormone called leptin, which works to promote satiety. This book, The Hungry Brain by Stephen Guyenet, is a great introduction to how this hormone works. In short, what we think of as appetite is not a singular entity. Multiple hormones are responsible for how hungry we feel at any given time. Leptin is a hormone secreted by fat cells, and the way it's supposed to work is that gaining fat increases the amount of leptin, which should reduce long-term hunger. Likewise, losing fat decreases the amount of leptin going to the brain, so long-term hunger should increase. In other words, if someone were to eat similarly to how they're already eating, but decrease portion size, the lower amount of leptin will ensure that this person's hunger increases, and they will end up back at their starting body fat percentage, despite their best efforts. So if obese individuals have a lot of fat cells, and therefore a lot of leptin, why are they still eating so much? The answer is because the brain becomes resistant to leptin signaling. Something about an unhealthy diet is causing inflammation in the hypothalamus, the area of the brain that regulates energy balance along with many other automatic processes, which is causing the leptin resistance. Also important is that the inflammation shows up before any other metabolic symptoms like obesity. That means depending on the quality of the diet, leptin resistance can change and appetite will reach a corresponding equilibrium. In that sense, the set point for fat storage is dynamic, and the term lipostat is used as an analogy for a thermostat. The amount of fat stored changes depending on the level of the brain's resistance to leptin, which depends on diet quality. Additionally, leptin signaling in the brain facilitates blood sugar control. To summarize, a nutritious diet prevents hypothalamic inflammation, which means the subject's leptin signaling works properly, keeping body fat levels normal and insulin sensitivity high. Conversely, a junk food diet causes hypothalamic inflammation, and the subject's leptin signaling works poorly, causing body fat levels to increase and insulin resistance to occur. The main question now is why junk food diets cause hypothalamic inflammation. We're going to dive into a fascinating new idea that makes a lot of sense for answering this. So we're focusing on an area of the hypothalamus called the arcuate nucleus, that pink part at the bottom labeled ARH. There are two types of brain cells there, or neurons, that help regulate eating behavior and energy usage: POMC neurons and AGRP neurons. The SF1 neurons in the ventromedial nucleus play a similar role as well, but for simplicity, let's mainly focus on AGRP neurons. One cool thing about mice studies is you can mess around with their genes. Remember how we talked about leptin signaling to the brain that the body has sufficient energy? Leptin normally helps suppress AGRP neuron activity because AGRP neuron activation promotes hunger and hunger-related adaptations, including insulin resistance. In this study published in the journal Nature, normal mice were compared with both mice who had no leptin receptors in AGRP neurons and no leptin receptors in the entire body. What was found is that the lack of leptin reception in this single cell type is responsible for most of the increased eating, weight gain, and insulin resistance we see when leptin signaling is not working properly. The paper on the right side, published in Cell, shows the neural circuitry between AGRP neurons and other parts of the hypothalamus for controlling both eating and blood glucose. Importantly, despite AGRP neuron activation causing both increased eating and blood glucose levels, the increased glucose happens in parallel with weight gain and is not a result of eating more food. A quick extra detail is that one of these pathways that leptin activates in the AGRP neuron is shared with insulin, which is why research shows some similar effects of blocking insulin in this neuron type. And the hypothalamic inflammation from junk food diets impairs insulin signaling in the hypothalamus in addition to impairing leptin signaling. But for simplicity, we'll simply refer to the sum of the resistance to these hormones as leptin resistance. Here's a great diagram demonstrating some of what happens with this impaired signaling. Remember, leptin comes from fat cells. If leptin signaling is dampened, the brain is fooled into thinking body fat is much lower than it actually is. With this falsely perceived starvation comes a multitude of energy conservation efforts. Less essential functions like reproduction are lowered, appetite skyrockets, and less energy is expended as heat via thermogenesis. Meanwhile, more glucose is released into the bloodstream from the liver, and glucose uptake into the muscle is prevented. That's because in a real starvation state, blood glucose must be prevented from dropping too low. During a false starvation from improper leptin signaling, blood glucose is already normal, so the extra glucose drives it above normal. The pancreas must then secrete more insulin to drive that glucose back down to normal levels, creating a persistent state of hyperinsulinemia. If metabolic health continues to decline and leptin signaling continues to get worse, energy conservation efforts can be pushed to the extreme and diabetes can develop, not to mention the suite of other chronic conditions that can develop along the way because of different cells conserving energy and not functioning optimally. A few additional effects of this energy conservation state are increased gut permeability, which can increase nutrient absorption at the cost of potential gut issues, fat around the organs as a more readily available fuel source than typical subcutaneous fat, increased blood pressure to maximize nutrient delivery, and alterations in food reward perception that helps incentivize food seeking. Remember, these are just a few of many adaptations that can cause noticeable effects on quality of life and daily function. To reinforce this point, we can look at lipodystrophy patients, people with rare genetic mutations preventing them from effectively putting on body fat. These patients are extremely lean, yet also have extremely high prevalence of metabolic problems like diabetes, excess food intake, and fatty liver. This is the exact opposite of what conventional ideas about body fat being the root of the problem would predict, but is perfectly explained by a central control model based around the hypothalamus and leptin. In fact, substantial improvements have repeatedly been shown by administering leptin to these patients, because although we've been discussing leptin resistance, a leptin deficiency, as is the case with lipodystrophy, functionally produces the same effects. The difference is that a case of deficiency can be treated with direct administration of leptin in pharmaceutical form with high amounts of success. So we know inflammation in the hypothalamus from some kind of mysterious damage leads to impaired leptin signaling. We know impaired leptin signaling leads to metabolic disease, but we still haven't answered what causes the hypothalamic inflammation in the first place. If we know the answer to that question, then we know what dietary patterns to adopt to stay healthy. And there's a very strong case to be made that the root of hypothalamic inflammation is something called oxidative stress. When energy balance neurons like AGRP undergo oxidative stress, hormonal signaling is dampened to prioritize the repair of those neurons, leading to leptin resistance. Similarly to a sprinter who rolls an ankle, at the cellular level, these neurons need to rest and repair when injured. Oxidative stress is centered around molecular oxygen wanting to steal electrons. This chemical feature, largely responsible for life on Earth as we know it, can also cause problems in the body. When a cell is making energy, electrons can sometimes leak and attach to oxygen prematurely. The extra unpaired electron on this oxygen makes it very reactive. This electron very much prefers to be paired, and this superoxide radical, as it's known, will happily steal an electron from a cell to stabilize itself, damaging that cell in the process. This radical is a part of a family of reactive damaging compounds called reactive oxygen species, or ROS. That being said, these ROS are not entirely bad. They perform various important roles in the body, like fighting pathogens and other signaling roles, but they become problematic when their concentration exceeds the body's ability to keep them in balance. Here's a quick view of how energy is produced in a cell via the electron transport chain. An electron is pushed through a series of protein complexes to pump hydrogen ions into the intermembrane space of the mitochondria. An enzyme called ATP synthase spins like a turbine, allowing those hydrogen ions to react with phosphate and ADP, creating ATP, the energy currency of the cell. Occasionally, shown in red, this process results in an electron leaking from one of the protein complexes. This is the sneaky electron that likes to attach itself to oxygen, creating a superoxide radical. Fortunately, our bodies have robust systems to handle these radicals. Any cell exposed to them has multiple lines of defense, the first being the water portion of the cell. First, the radical is reduced by an enzyme called superoxide dismutase, SOD, into hydrogen peroxide, H2O2. Although hydrogen peroxide is not a radical, it still falls under the category of reactive oxygen species mentioned earlier. It can still react with and damage cells, but it is much less reactive than a radical. H2O2 then reacts with other enzymes, either glutathione peroxidase or catalase, to become harmless and stable water. But if H2O2 is not reduced to water, it can react with iron or another superoxide to form a hydroxyl radical, HO, for which no known scavenger exists. If ROS gets past the water portion of the cell, those ROS can damage the fats, or lipids, inside a cell in a process called lipid peroxidation. Before understanding lipid peroxidation, we should review how different types of fats are categorized from a chemical standpoint. We have saturated fats, characterized by 0 carbon-to-carbon double bonds, monounsaturated fats, which have 1 double bond, and polyunsaturated fats, which have multiple double bonds. The multiple double bonds in polyunsaturated fats are uniquely susceptible to lipid peroxidation due to the fragility of the carbon in between the double bonds. Here's a big scary diagram showing what happens with lipid peroxidation. Notice the upper left of the diagram is the water portion of the cell we looked at earlier. We can see how it fits into this bigger picture. We can first have various radical sources, whether they're hydroxyl or superoxide from insufficient defense in the water portion of the cell, or xenobiotics, which is a fancy way of saying some external chemical like an environmental pollutant. Those react with lipid membranes in the cell to create lipid radicals and lipid peroxyl radicals. Those new lipid peroxyl radicals can once again react with the lipids to create new lipid radicals in a chain reaction. Luckily, glutathione steps in again to reduce those new radicals and create stable lipids and lipid hydroperoxides. Glutathione peroxidase helps stabilize those remaining lipid hydroperoxides. Meanwhile, vitamins C and E also help to stabilize lipid peroxyl radicals. All these factors together help to blunt the chain reaction and minimize damage. We can apply this idea, practically speaking, to the debate on seed oils, more commonly known as vegetable oils for marketing reasons. There's a growing idea that the abundance of these oils in ultra-processed foods is contributing to disease. The biochemical evidence we just reviewed strongly supports this argument. Remember, polyunsaturated fats are uniquely susceptible to lipid peroxidation, and these oils are an unstable extract of mostly polyunsaturated fat. That means these oils are much more susceptible to lipid peroxidation than whole food sources of these fats. Heating these oils oxidizes them, leading to a breakdown in their fatty acids and creating extremely reactive aldehydes. Once ingested, these aldehydes can damage cells. Glutathione, our endogenous antioxidant hero, has some ability to help detoxify these aldehydes, but avoiding this chain of events as much as possible in the first place is the ideal way to maintain cellular health. Okay, we know oxidative stress is bad, but why is it leading to metabolic problems? First, there are several reasons that the brain is highly susceptible to oxidative stress. The hypothalamus, which is the part that controls energy balance, is one of the most, if not the most susceptible regions out of this one big susceptible organ. Those energy balance neurons, like AGRP, work hard to produce energy and direct metabolism when food is ingested. When a lot of ATP is created in these neurons via the electron transport chain, a lot of electrons can leak in that process and easily create oxidative stress in this weakly defended environment. So it's that exact oxidative stress that is damaging energy balance neurons, making them resistant to leptin, and causing all the hallmark symptoms of metabolic disease. That's why it's not calorie tracking that matters, but food quality. Proper nutrient intake ensures that energy balance neurons are defended against stress and remain sensitive to leptin, so that body fat, appetite, and insulin sensitivity are managed automatically. Now that we understand the problem and solution from a theoretical standpoint, we can look at the importance of various nutrients to keep antioxidant defense strong, nutrients that are often sorely lacking in a standard Western diet. Glutathione is an extremely important antioxidant that shows up in many stages of antioxidant defense. We want to make sure our cells have sufficient glutathione, so it helps to know how it's made. Glutathione is synthesized from the building blocks of protein called amino acids, specifically the amino acids cysteine, glutamate, and glycine in a two-step reaction. That's why consuming an abundance of protein with these amino acids is crucial for antioxidant defense, and why diets rich in protein have such favorable results on health compared with diets lower in protein. Looking at some vitamins and minerals, we can see copper, zinc, and manganese are important for superoxide dismutase, selenium, which along with glutathione is contained in glutathione peroxidase, iron and manganese are used for catalase, vitamin E and vitamin C help reduce lipid peroxyl radicals, vitamin C also helps as a radical scavenger in general, and B vitamins help recycle glutathione. This is only scratching the surface, but we can see how these different nutrients play different roles in a multifaceted antioxidant defense. Worth noting about vitamin E is that its only known role in the entire body is stabilizing lipid peroxyl radicals, so the amount required depends on the amount of polyunsaturated fat consumed. Those consuming diets lower in polyunsaturated fats require less vitamin E. It's one example of how nutrient requirements can vary from person to person, and a static recommended daily allowance is not a catch-all. There's also a lot of non-essential antioxidant supportive nutrients that are often overlooked. Amino acids like taurine, carnosine, carnitine, and anserine all fall into this category, as well as coenzyme Q10. Popular culture attributes antioxidants to plants, but the evidence for how important essential amino acids are to glutathione synthesis, as well as non-essential amino acids are as direct antioxidant defenders, demonstrates how powerful animal-based foods are to maintaining a healthy metabolism. Even polyphenols, which are plant compounds that can assist with antioxidant defense in different ways, are contained in animal foods in addition to plant foods. So while plant foods can have useful nutrients, the pop culture view of plants as the superheroes of health is greatly exaggerated. Let's look at polyphenols in a bit more detail. There are so many of these that exist. The number of identified polyphenols is in the thousands, and they can be bioactive radical scavengers, do absolutely nothing, or somewhere in between. But it does offer a compelling answer to why research overwhelmingly supports honey as preferable to refined sugar for health. Honey contains a variety of bioactive polyphenolic compounds and has been shown to increase oxygen radical absorbance capacity. This feature manifests itself as protective against metabolic syndrome, especially when compared with refined sugars. Regardless of opinions on whether honey consumption should be limited or not, what is clear is that consuming honey is a much better alternative to consuming refined sugars. When stressed, cells will naturally boost the synthesis of enzymes that facilitate antioxidant defense. The way this is done is via a transcription factor called NRF2, which binds to DNA sequences and allows this instruction to take place. Small stressors can have an effect where the resiliency created through NRF2 activation outweighs the stress itself, an effect called hormesis. Some polyphenols are technically pro-oxidant, but are called antioxidants because they induce NRF2 activation. Other stressors like exercise and sauna use can also stimulate NRF2 hormetically. Note that NRF2 primarily boosts the production of enzymes, but not the substrates needed for those enzymatic reactions. For example, an increase in an enzyme like glutamylcysteine ligase is only useful if there's enough glutamate and cysteine. A shortage of these amino acids means a limitation in this part of the reaction, limiting potential glutathione synthesis. That's why good nutrition still needs to be considered when hormetic strategies are used. Smoking can directly cause diabetes, according to the CDC. The way this happens is more peripheral than centralized. Nicotine downregulates NRF2, weakening cellular responses to stress and leading to insulin resistance. It's another way that insulin resistance can develop independent of food intake. Here's a summary of the important points and a few more studies supporting the main idea. We can define a metabolically unsound diet as one that creates an oxidative burden above what antioxidant defense systems can handle, especially in the brain's hypothalamus. The resulting damage impairs cellular signaling from critical hormones, mainly leptin, in a way that the brain is fooled into thinking the body is in a starvation state. Insulin resistance, excess appetite, and other maladaptations to conserve and store energy occur to protect against this false starvation. These energy conservation efforts can also lead to suboptimal cellular function in various parts of the body, contributing to diseases that may appear unrelated. I want to highlight one study to challenge the well-intentioned but misinformed idea that carbohydrates directly damage and inflame cells. In this human study, large amounts of glucose were injected and left circulating for a few hours, assisted with an insulin-blocking drug. Inflammatory markers were measured to assess the amount of damage the glucose caused in this short time. An experimental group that received glutathione injections were compared with a control group. Based on what we've been talking about, you might predict inflammation in the glutathione group to increase less than in the control group. Well, not only was inflammation in the glutathione group less, it was zero. That's right. The glutathione group had zero increase in inflammatory markers from start to finish, while the control group had a significant increase. It's a perfect demonstration of how inflammation in a metabolic context is caused and how it can be remedied. The cause is simply excess ROS generation from ATP production and can be avoided entirely by ensuring proper antioxidant defense. A standard American diet, being high in unstable refined oils, high in nutrient-poor sources of carbohydrates, and high in chemical additives, is a perfect recipe for disaster. Calories are used as a scapegoat when in fact there is nothing wrong with calories per se. Eating high-calorie foods is delicious and healthy when those calories come with an abundance of nutrients to support antioxidant defense. That's why 1,000 calories of steak beats 1,000 calories of cake every time. Finally, we can speculate why this adaptive mechanism may have been useful in a naturalistic context. This book, called Nature Wants Us to Be Fat, is good material to help think along those lines. It talks about how different animals prepare for food scarcity, which might be why oxidative stress promotes energy conservation in the first place, as an anticipation of upcoming food shortage. The book focuses on fructose, and the research and conclusions drawn leave much to be desired in my opinion, but it helps with this kind of thinking, even if the context is different than the material we're covering here. Perhaps foods in nature that were high in energy and relatively low in antioxidant defense were abundant before food-scarce seasons in some regions, creating an evolutionary adaptation in some ancient mammalian ancestor that persists in humans and other mammals today. It's a speculative idea, but fun to think about why things are the way they are. That's all I have here, but you can read my blog to see how these ideas can help solve other health mysteries that continue to stump everyone. The reference papers from this presentation are in the description.